Hello guys, welcome. Welcome to Ashutosh Coaching Center. Now today we are going to start chapter number 6, and the name of chapter is Basic of Molecular Basis of Inheritance. Let's start. पता नहीं आवाज को आज क्या हो गया, चलिए कोई नहीं, I'll manage. In the previous chapter you have learned that the inheritance and the pattern, okay, and the genetic basis of such patterns at the time of Mendel, and the nature of those factors and regulating the patient of the inheritance was not clear. Over the next 100 years, the nature of putative genetic material was investigated, and the culminating in the realization that the DNA deoxyribonucleic acid and is the genetic materials and at the least for the majority of organisms. In class 11th, you have learned that the nucleic acid are polymers of nucleus. Deoxyribonucleic acid DNA and ribonucleic acid RNA are two types of nucleic acid are found in the living system. DNA acts as the genetic material in the most of the organism. RNA also act as a genetic material and in some viruses mostly function as a messenger. RNA as an additional role as well, and it functions as chapter and it functions as adapter structurals and in some case as a catalytic molecules. In class 11th you have already learned the structure of nucleotide and the way these monomer unit are linked to formed nucleic acid polymers. In this chapter we are going to discuss the structure of DNA and its replication, the process of making RNA and DNA transcription, okay, and genetic code. देखो एक बात ध्यान रखना, डीएनए का बनना ट्रांसक्रिप्शन कहलाता है, ओके, and genetic code determines the sequence of amino acid in proteins. The process of protein synthesis मतलब प्रोटीन का बनना क्या कहलाता है, ट्रांसलेशन, ओके, यानी कि राइबोसोम में बनता है, ओके, एंड ओके the elementary basis of their regulation, the determination and the determination, okay. थोड़ा सा आवाज का थोड़ा दिक्कत हो रहा होगा बट मैनेज करना पड़ेगा ना. हाँ, of nucleic as of complete nucleotide sequence of humans genomes during the decade and has set in a new era of genomic. And in the last section the essential of the human genome and the sequencing and its consequence will also be discussed. Let us begin our discussion by the first understanding the structure and the most interesting molecules in the living system, that is the DNA. In the subsequent section we will understand that is why is the most abundant genetic material and what its relationship is with RNA. 6.1 DNA. DNA is a long polymer deoxyribonucleic acid nucleotides, okay, and the length of DNA is usually defined as the number of nucleotide or a number of polynucleotide refers to as a base pair and present in this also is the characteristic organism and for the organism. And for example, and a bacteriophage known as the 5 into 10 174 has 5386 nucleotide, and the bacteriophage lambda has 48502 base pair BP, and the Escherichia coli has 1.6 into 10 to the power 6 BP, and haploid content of DNA is 3.3 into 10 to the power 9 BP. Let us discuss the structure of such a long polymer. 6.1.1 structure of polynucleotide chain. Let us recapitulate recapu, sorry, let us recapitulate and the chemical structure of polynucleotide chain, DNA or RNA. The nucleotide has three components: a nitrogen base and a pentose sugar, ribosome in the case of RNA, and deoxyribose of DNA, and a phosphate group, okay. And there are two type of nitrogenous base, प्यूरिन्स एंड एडेनिन्स एंड ग्वानिन्स। ओके, तो इसमें दो टाइप के नाइट्रोजन बेस होते हैं। प्यूरिन्स होता है, प्यूरिन्स में क्या होते हैं, एडेनिन एंड ग्वानिन। ओके, एंड द पिरामिड्स साइटोनिन यूरासिल, काफ़ी इंपॉर्टेंट लाइन है। हाँ, pyramid pyrimidines and the cytosine uracil thymine, and the cytosine is the common for both DNA and RNA, and thymine is present in the RNA, and uracil is present at the place of thymine, and nitrogenous base is linked to the OH of 1 degree carbon or pentose sugar through a N glycosidic linkage to a form of nucleoside such as the adenosine or deoxyadenosine and guanosine and deoxyguanosine and the cytidine and the cytidine and or deoxycytidine or uridine of deoxythymidine. When a phosphate group is linked to OH of 5 carbon nucleotide through the phosphaster and linkage, a corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Two nucleotides are linked through 3 to 5 phosphate digester linkage to form a nucleotide. More nucleotide can be joined in such a manner to form nucleotide chain. A polymer thus formed has at one end a free. This is the structure. You should remind this. Okay, आपको ये चीज़ को याद कर लेना है, चाहे जैसे भी हो सके इसे याद करना है तो करना है, क्योंकि ये एग्ज़ाम के पॉइंट ऑफ व्यू से काफ़ी ज़्यादा पूछे जा सकते हैं। हालांकि जाते का मैटर नहीं करता, लेकिन जा सकते हैं इसका भी कोई गारंटी नहीं है। हमारा काम है एनसीईआरटी पूरा ख़त्म करना जो हम कर रहे हैं। द फॉस्फेट मोइटी एट 5 the end of sugar 5 dash end of the sugar, which is referred to as the 5 dash end of the polynucleotide chain. Similarly, at the other end of polymers, the sugar and has free OH of 3 carbon group, which is referred to as the 3 end of the polynucleotide chain. And the backbone of polynucleotide chain is formed and due to the sugar and the phosphate. The nitrogenous base linked to the sugar and the moiety project from the backbone 6.1. In RNA, every nucleotide residue has additional OH group present at two position in the ribose. Also in RNA uracil is found at the place of thymine and the 5-methyl uracil, and another chemical name is thymine. And DNA as an acidic substance present in the nucleus was identified the Frederick Meeker in 1869. His name is nuclein, and however, due to the technical limitation in isolating such a long polymer intact, and the education and elucidation of a structured DNA remains exclusive for a very long period of time. It was only in 1953 James Watson and the Francis Crick, based on X-ray diffraction data produced by the Maury Wilson and the Rosalind Franklin, and the proposed by very simple and but a famous doll helix model for the structure of DNA. One of the hallmarks for their proposition and was base pairing and uh, base pairing between two strands of polynucleotide chain. Okay, however, this proposition was also based on the observation of Erwin Chargaff that for a double stranded DNA and the ratio between the adenine, thymine and the guanine and cytosine are the constant and equal one. The base pairing confers a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore if the sequence of a base in one strand is known, then the sequence in other strands can be predicted. Also, if each strands from the DNA, let us call it as parental DNA, act as a template for synthesis of a new strand, and the two double-stranded DNA. Let us call them as a daughter DNA. Thus, produced would be identical to the parental DNA molecules. Because of this, genetic implication of structure of DNA became clear. The salient feature of the double helix structure of DNA as follow. It is made of two polynucleotide chain where the backbone is constituted by the sugar phosphate and the bases project inside. The two chain have anti-parallel polarity. It means if one of the chain has polarity 5 to 6 and the other has 3 to 5, okay, and the bases in two strands are paired through a hydrogen bond and H bond forming base pair BP adenine forms two hydrogen and H bond thymine and from the opposite strand and vice versa. Similarly, guanine is bonded to site with the cytosine and with a 3H bonds. As a result, always purine comes opposite to a pyrimidine and which generated approximately uniform distance between the two strands of the helix figure 6.2. And fourth number में दे रखा है हमारा the two chains are coiled in a right handle fashion. The pitch of the helix is 3.4 nanometer. A nanometer is a billionth of a meter into 10 to the power minus 9. Okay, it means there are roughly and 10 bp in each. इस चीज़ को आप भी याद कर लीजिए। सारे जितने भी डायग्राम दिख रहे हैं इसको आप अलग पेज में ड्रॉ ही कर लें, क्योंकि एनसीईआरटी तो पढ़ते पढ़ते रहेंगे बट इसको थोड़ा ध्यान रखना भी जरूरी है। नाउ and turns consequently the distance between a BP in a helix is approximately 0.34 nm. इससे को आप याद रखिए तो ज़्यादा बेटर हो सकता है। ओके, तो BP कितना होता है अप्रोक्सीमेटली 0.34 nm कह सकते हो। The plane of one base pair stack over the other in double helix. This is the addition of H bonds and the coniferous stability of helical structure. Figure 6.3. Compare the structure of purines and the pyrimidine. Can you find out why the distance between the polynucleotide and chain in a DNA remains almost constant? The proposition of the double helix structure of DNA for DNA and its simplicity in explaining the genetic implication became revolutionary, and very soon Francis Crick proposed the central dogma in molecular biology, okay, and which states that the genetic information flows from the DNA to RNA to protein, okay. हाँ, इस चीज़ को आप ध्यान में रख के समझ जाए। सेंट्रल डोग्मा क्या है, तो इसमें प्रोसेस होता है ये कंप्लीट, ओके। इसमें क्या होता है, डीएनए से अगर डीएनए बन रहा है, डीएनए रेप्लिकेट कर रहा है, तो उसको हम लोग बोलते हैं रेप्लिकेशन, जो कि आपको यहाँ सीधे सीधे यहाँ पे दिख सकता है, देयर इज, इसको कहते हैं रेप्लिकेशन। और अगर यही डीएनए से बन जाए आरएनए तो हम उसको बोल देंगे ट्रांसक्रिप्शन, ओके, ट्रांसक्रिप्शन। और अगर यही जो एमआरएनए से अगर ये बनता है प्रोटीन तो इसको हम लोग कहते हैं ट्रांसलेशन, ओके। हाँ, ये बात को ध्यान रखें, और से इसका फोटो खींचे तो भी बेटर होगा आपके लिए, क्योंकि काफ़ी ये एग्जाम पॉइंट से आ सकता है। हाँ, इस चीज़ का भी आप पहले ही फ़ोटो ले लो तो ज़्यादा बेटर होगा मेरे ख्याल से। सॉरी, ये फ़ोटो में एन ही कट गया था, पूरा नाम आना चाहिए था ना। यस, नाउ इट्स ओके, नाउ इट्स डन, ओके। सो आते हैं अपने आगे वाले लाइन पे। सो गाइज, आप ध्यान से ये बदला भूले कि इसको चैनल को और भी लोगों को तक पहुंचाना है, जिस कारण से ताकि उसे भी एनसीईआरटी का जो ऑनलाइंस जो होते हैं वो रीड करता हुआ एक मिल जाए, या मतलब कह सकते हो कि एनसीईआरटी रीडिंग का ये प्लेलिस्ट उसी पे मिल सके, ताकि उसके भी बाद में बहुत काम आ सके। सो जितना हो सके इस चीज़ को शेयर करें ताकि हमें भी इंटरेस्ट आए। बाकी तो आप लोग पे है, आप क्या करते नहीं करते वो तो आप ही जानेंगे। तो स्टार्ट करते हैं। 6.1.2 ओके, पैकिंग ऑफ डीएनए हेलिक्स taken the distance between two consecutive base pair of as 0.3 nm. It means 0.3 into 10 का power minus 9 मीटर। अब वैसे 10 का पावर माइनस 9 को 10 इनवर्स 9 भी कहते हैं। ओके, ये बात ध्यान रखना। If the length of DNA double helix is typical mammalian, is cell is calculated simply by the multiplying total number of BP and distance between the two consecutive BP. That is 6.6 into 10 के power 6 BP into 0.034 into 10 inverse इतना B per BP. It comes out approximately 2.2 meters and length, that is far greater than dimension under typical nucleus, approximately 10 inverse 6 meter. How is such a long polymers packaged in a cell? If length of E. coli DNA is 1.36 mm, and can you calculate it the number of सॉरी ये गलती से हो गया, हमें इसका स्क्रीनशॉट लेना है अंडरलाइन करते हुए ताकि आगे जाके काम आ सके। हाँ, हो गया। अब बात कर रहा है कैन द नंबर ऑफ बेस इज कैन द नंबर ऑफ बेस पेयर इन ई कोलाई, मतलब क्या आप कैन यू कैलकुलेट, क्या आप कैलकुलेट कर सकते हैं नंबर? तो हमको से मतलब तो घंटा नहीं है, आप वो देख लीजिए, अगर वो थोड़ा इंटरेस्ट हो तो बता दीजिएगा। हाँ इन प्रोकैरियोटिक्स सच एज ई डॉट कोलाई, through they do not have defined nucleus. The DNA is scattered throughout the cell. DNA being negatively charged and is held with some proteins have positive charge. ओके, ये बात ध्यान रखें। डीएनए जो होता है उस पर निगेटिव चार्ज होता है और प्रोटीन जो होता है उस पर पॉजिटिव चार्ज होता है। इन अ रीजन termed as nucleoid and the DNA is in nucleoid is organized in large loop held by proteins. In eukaryotic, this organization is more complex, and there is a set of positively charged basic protein called histone. ओके, हिस्टोन प्रोटीन क्या होता है ये सब जान लिए। मतलब उसमें ऑर्गेनाइज एक पॉजिटिव प्रोटीन होता है, मतलब पॉजिटिव जो होता है ना, पॉजिटिव क्या कर सकते हैं, पॉजिटिव चार्ज प्रोटीन जो होता है उसको हम लोग हिस्टोन प्रोटीन भी कहते हैं। The protein acquired charge depending on upon the abundance of amino acid residues with charged and the side chain. Histone are rich in the basic of amino acid and residue lysine and arginine, and both the amino acid residue and the carry positive charge in their side chain. Histones are the organized to form the unit of 8 molecules of the histone octamer. Okay. Stone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer is to form a nucleosome. Okay, तो बाद में हिस्टोन ऑक्टेमर में ही जाके बाद में न्यूक्लियोसोम बनता है। ओके, और आगे देख लिया जाए। हाँ, तो नेगेटिवली चार्ज ऑफ डीएनए इज रैप्ड अराउंड द पॉजिटिवली चार्ज polarly charged and stone octamers to form structural is called nucleosome. Typical nucleosome contain 200 bp of DNA helix, and nucleosome constitutes the repeating unit of a structure of in a nucleus called chromatin. ओके, तो क्रोमेटिन क्या होता है ये भी जान लेना चाहिए हम लोग को, तो जान लिए है। और द थ्रेड like stained colored bodies in the nucleus. The nucleus chromatin as seen in are seen as beating beads on string. Structure when viewed under the electron microscope. EM figure 6.0. Theoretically, how many base nucleosome do you imagine present in a mammalian cell? The baked on strings are structured in chromatin in package to form chromatin fibers that are further coiled and the condensed metaphor stages and of cell to form chromosome and the packaging of chromatin at higher level and acquired additional set of proteins that collectively are referred to as non-histone. Okay, that refers to as non-histone chromosomal protein. In a typical nucleus, some regions sons of chromatin are loosely packed. Okay, loosely packed and stain light are referred to Eurochromatin. The chromatin that is more densely packed and the stained dark are called heterochromatin. Okay, Eurochromatin is said to be transcriptionally and active chromatin, whereas the heterochromatin is inactive. 6.2 The search of genetic material. Even though the discovery of nuclein by the Mesker and the proposition of for the principle of inheritance by the Mendel were almost at the same time, but that the DNA act as the genetic material look took the long to the discovered and the proven. By 1926, the quest to determine the mechanism for the genetic inheritance had reached the molecular level, and the previous discoveries by the George Mendel and the Walter Simon and the Thomas Hunt Morgan and the numerous other scientists. Okay, and had narrow had to narrow search to the chromosome located in the nucleus of most soil, but the question of what molecules was actually genetic material and had been answered transforming principle in 1928. The Frederick gifted in a series of experiments with streptococcus pneumoniae and the bacterium responsible for the pneumonia witnessed miraculous and the transformation in the bacteria. During the course of his experiment, a living organism bacteria has changed in physical form. When Streptococcus pneumonia and Pneumococcus bacteria are grown on a culture plate, some produce smooth shiny colony as while other produces through a colony R. This is the because as a strain bacteria have a mucus polysaccharide coat and while the R strain does not, mice if infected with the S strain virulent died from the pneumonia infection, but mice affected the ratio and the R strains do not develop the pneumonia. R strains injected to a mice and mice die. R strain injected dies mice live. Okay, काफी important line है ये सब. Griffith was able to kill bacteria by the heating them, and he observed that heat killed strain bacteria inject to the mice and did not kill them. He as strain bacteria injected to mice live, and as strain plus heat plus R strain inject mice die. Injected a mixture of heat killed S and lives are bacteria. The mice dead. Moreover, he discovered living as bacteria from the dead mice. He concluded that the R strain bacteria and somehow been transferred by the heat killed S strain bacteria. Some transforming principle transferred from the heat killed S strain and had enabled the R strains to synthesize a smooth polysaccharide coat and become a virulent. This must be due to the transfer of the genetic material and however the become biochemical nature of genetic material was not defined from his experiments. Biochemical characterization of transferring principle. Prior to the work of Oswald Arbery and the Colin MacLeod and Malkin McCarthy, 1933 to 1944, and to genetic material was vouched to be a problem, and they worked to determine the biochemical nature of transforming principle to Griffith's experiment. They verified biochemicals, proteins, DNA, RNA, etc. from the heat-killed S cell to see which ones could transfer live R cell into S cell. Okay, इसमें याद रखना ये भी लाइन बहुत इंपॉर्टेंट है, आर सेल से एस सेल बनता है, ठीक है, आर सेल एस सेल वेरी इंपॉर्टेंट रहेगा. एंड दे डिस्कवर्ड डीएनए alone as bacteria and caused R bacteria because R bacteria to become transferred. They also discovered that the protein digesting enzymes protease and देखो ये भी रखना, जो प्रोटीन डाइजेस्ट करता है उसको लोग प्रोटीएज कहते हैं, ठीक है ना. हाँ. And RNA digestion enzymes RNAs did not affect the transformation, show the transforming substance was not a problem, or DNA proteins or DNA digestion and with DNAs did inhibits transformation, suggestion that the DNA caused the transformation, and they concluded that the DNA is the hereditary material, but not all the biologists were convinced. Can you think of any differences between the DNAs and DNAs? Yes, maybe it is. 6.2.1, the genetic material is DNA, and the unquestionable proof that the DNA is the genetic material came from the came from the experiment of Alfred Hershey and the Maratha's case 1952. They worked with the viruses that infect in virus bacteria and they called bacteriophages. And the bacteriophages attaches to the bacteria and its genetic material in that enter the bacterial cell, and the bacterial cell treat the viral genetic materials as if it was its own and subsequently manufacture more virus particles. Okay, and the Hershey and the Chase worked through the discovered whether it was protein or the DNA from the viruses that entered the bacteria, and they grew some viruses on a medium that contained radioactivity phosphorus and some on the medium that contained radioactivity and sulfur. Virus grown in the presence of radioactivity and the phosphorus contained radioactivity DNA, but not radioactive protein because DNA contain phosphorus but protein does not. Similarly, virus grown on radioactivity sulfur contain radioactive protein but not radioactive DNA because DNA does not contain sulfur. Now, radioactive phase were allowed to attach E. coli bacteria. Then, as the infection proceeded, the viral coat were removed from the bacteria by the agitating them in a blender. The virus particles were separated from the bacteria by the spinning them in a centrifuge. Bacteria which was infected with virus had radioactive DNA and were radioactive, indicating that DNA was material that passed from the virus to the bacteria, and the bacteria that were infected with viruses that had radioactive protein were not radioactive. This indicates that protein did not enter bacteria from the viruses. DNA is the therefore genetic material that is the passed from the viruses to bacteria. आप इस मतलब डायग्राम को भी नोट कर सकते हैं, काफ़ी एग्जाम के पॉइंट ऑफ व्यू से काफ़ी इंपॉर्टेंट हो सकता है. ओके, इसको भी स्क्रीनशॉट ले लिया जाए, बेटर होगा. यहाँ पे देखिए ये बैक्टीरिया फेजेस है और ये बैक्टीरिया रेडियोएक्टिव 32P है, जबकि कहने की रेडियो एक्टिविटी 35P लेवल जो होता है वो प्रोटीन कैप्सूल कहलाता है. ओके, अब आगे देख लेते हैं, ये सबको तो आप नोट कर लो, ज़्यादा बेटर होगा मेरे ख्याल से. ये हमारा हार्शी चेज एक्सपेरिमेंट भी कहलाता है, ठीक है. अब आगे देखते हैं. 6.2.2 द प्रॉपर्टीज ऑफ जेनेटिक मटेरियल डीएनए वायरस वर्सेस versus RNA. From the from the foregoing discussion, it is clear that the debate between the protein versus DNA as the genetic material was the unequivocally resolved from the Hershey-Chase experiment. It became an established fact at its DNA that act as a genetic material. However, it subsequently became clear that uh, in some viruses, RNA is the genetic material. For example, tobacco mosaic viruses and the cubic bacteriophage etc. Answer to some of the questions such as why the DNA is predominant genetic material and the various RNA performs dynamic function of messenger and adapter has to be found from the differences between the chemical structure of the two nuclei and nucleic acid and molecules. Can you recall the two chemical differences between the DNA and RNA? A molecule that can be act as a genetic material but must fulfill the following criteria. First, it should be able to generate its replica and replication, okay, and it should be stable chemically and structurally. Third, it should provide the scope of for slow changes and mutation that are required for evolution. Fourth, it should be able to express and itself it in the form of Mendelian character. उसके बाद है हमारा if one examine each requires one by one because of rule of base and the pairing of complementary and both the nucleic acid and DNA RNA and have been ability to direct their duplication and the other molecules in the living system such as a protein fail in the fulfill first criteria itself, and genetic material should be stable enough to change with different stage of life cycle age or the change in the physiological of the organism. Stability as the one of the properties of genetic material was where is evidence in grafts and the transforming principle itself that heat which killed bacteria and at least did not destroy the sum of the properties of genetic material, and this can be now easily explained like in the light of DNA that two strands being complementary it's if separated by the heating come together when appropriate conditions are provided for the 2OH group present at every nucleotide is that RNA is a radioactive group which makes RNA label and it's degradable, and RNA is also known to be catalytic, okay, hence the catalytic. Therefore DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore among the two nucleic acid the DNA is better than genetic material, okay, and in fact the presence of thymine at the place of uracil also confers additional stability of to DNA. Detailed discussion about the required understanding of the process of repair of DNA and that you will be study these process in higher classes. Both DNA and RNA to mutate. In fact, RNA being unstable mutate to at as a faster rate. Consequently, viruses having genomes and having shorter lifespan and mutate and evolve faster. RNA can directly decode code for the synthesis of protein and hence can easily express the character. DNA, however, it is the dependent on RNA for synthesis of protein, and the protein synthesizing machinery has evolved around RNA. And above the discussion indicate that both RNA and DNA can be function as function and genetic material, but DNA being more stable preferred for the storage of genetic information. For the transmission of genetic information, RNA is better. Our 6.3 RNA world. From foregoing discussion and immediate questions becomes evident which is the first genetic material. It shall be discussed in detail chapter on chemical evolution, and but briefly we shall highlight some of the fact and points. RNA was the first genetic material, and there is enough evidence to the suggested that the essential life such as metabolism, translation, and splicing, etc. Evolved the RNA, and the RNA used to act as a genetic material and as a as well as catalytic, and there are some important biochemical reaction in the living system that are catalyzed by the RNA catalytic and not by the protein enzyme, but RNA beings catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that make it forced more stable. DNA being double-stranded and having complementary strand further resist the changes by the evolving process of repair. 6.4 replication. While the proposing the double helical structure for DNA, Watson and Crick immediately proposed a scheme of DNA. Okay, तो वॉटसन एंड क्रिक जो थे उन्होंने ही डीएनए का स्कीम जो था वो प्रपोज किया था. ओके, एंड द क्वाट and to quote their original statement, and that is follow as it has not escaped our notice that specific repairing we have to postulate immediately suggested a possible copying and mechanism for the genetic material, and Watson and Crick 1953. Okay, the scheme suggested that the two strands would separate act as a template for the synthesis of a new complementary strand. After the completion and of replication, each DNA molecules would have one of the parental and one newly synthesized strand. This scheme was termed as semi-conservative DNA and replication. Okay. Hmm. उसके 6.4.1 the experimental proof. It is now proven that the DNA replicate and the semi conservatively. It was shown in first Escherichia coli and subsequently in higher organisms such as plant. Okay, and human cell. Matthew Michelson and the Franklin Stahl performed the following experiment in 1958. They grew E. coli in a medium containing 15 NH4 Cl 15 nitrogen. 15N is the heavy isotope of nitrogen. As the only nitrogen source of many generations, the result was that 15N was incorporated in the newly synthesized DNA. Okay, न्यूली सिंथेसाइज डीएनए. यार ये क्या हो रहा है, इसका स्क्रीनशॉट लेना जरूरी है. आप भी लोग इसका स्क्रीनशॉट ले लें या फिर इसको नोट कर लिए, काफ़ी इंपॉर्टेंट लाइन है. हाँ, okay, synthesized DNA and as well as other nitrogen containing compound. This heavy DNA molecules could be distinguished from the normal DNA by the centrifugation in a caesium chloride and CSCl density gradient. Okay, इसका भी आप फार्मूला को याद कर ले, मे बी एग्जाम में आ भी जाए. ओके, हाँ, ये भी जाने. प्लीज नोट दैट 15 nitrogen is not as a radioactive isotope, but it can be separate from 14 on nitrogen only based on densities. Okay, and then they transferred the cell into a medium with normal 14 NH4 Cl and took sample at a various definite time interval as the cell multiplied, and extracted the DNA that remains as a double standard helix, and the various sampled were separated independently on CSCl gradient to measure the density of DNA. Can you recall what the centrifugal force is and think why the molecules with higher mass density would sediment faster? इसका भी आप स्क्रीनशॉट ले लो तो मेरे ख्याल से ज़्यादा बेटर होगा, बिकॉज ये सब एग्ज़ाम के पॉइंट ऑफ व्यू से ज़्यादा इम्पोर्टेंट है. उसके बाद हमारा 10 DNA was not extracted from the culture one generation after the generation from the 15th and to 14th and medium that is after 25 minutes. The E. coli divides into 20 minutes and a hybrid and had a hybrid of or intermediate density. DNA extracted from the culture after another generation that is after 40 minutes. Okay, एंड द सेकेंड जनरेशन. इस ये पूरा जो लाइन वो काफ़ी ज़्यादा ही इंपॉर्टेंट है. इस लाइन को ध्यान में रख ले आप लोग, ज़्यादा बेटर होगा. इस लाइन को भी नोट कर ले. अब आगे जाते हैं. कंपोज्ड ऑफ इक्वल अमाउंट ऑफ hybrid DNA and light DNA. Okay, now if E. coli was allowed to grow for 80 minutes, and then what would be proportion of light and hybrid density molecule? Very similar experiment involving the use of radioactive thymidine to detect the distribution of newly synthesized DNA is in the chromosome was performed on Vicia faba, and the faba means by the Taylor and the Collegiu in 1958. The experiment proved that the DNA in the chromosome are also replicate and semi-conservatively. 6.4.2 Machinery and enzymes. In living cells such as E. coli, the process of replication requires a set of catalytic enzymes, and the main enzymes is referred to as DNA depend on the DNA polymerase. Since it uses DNA templates to catalyze and the polymerization of deoxynucleotide. These enzymes are highly efficient enzymes as they have catalyzed and the polymerization of a large number of nucleotide in a short time. E. coli that only 4.6 into 10 power 3 BP compared with the humans whose diploid content is 6.6 into 10 power 9 BP complete the process of replication within 18 minutes, and that means the average rate of polymerization has to be approximately 2000 bp per second. Not only do these polymerase have to be fast, and but they have to catalyze the reaction with degree of accuracy. Any mistake during the replication would result into mutation. Furthermore, energetically replication is a very expensive process, and deoxyribonuclease triphosphate serve dual purposes, and in addition to acting as substrate, but they provide energy from the polymerization reaction. The two terminal phosphate in a deoxyribonucleic nucleoside and the triphosphate are high energy phosphate, and same as in the case of ATP. Okay, in addition of DNA polymerase, and the many additional enzymes are required to complete the process of application of high degree of accuracy for long molecules, and since two strands DNA cannot be separated and is its entire length due to a high energy requirement, the replication occurred within a small opening of the DNA helix referred to as a replication fork. The DNA dependent DNA polymerase catalyzes polymerization and only in the one direction, and that is 5 to 3, and the creates some additional complication at the replication fork, and consequently one strands and the template with a polarity 3 to 5. The replication is continuous while the other template with the 5 to 3 and it is discontinuous synthesized fragments are later joined by the enzymes and DNA lipase. Figure 6.8, and the DNA polymerase on their on cannot initiate the process of replication. Also, the replication does not initiate randomly at any places in DNA. There is a definite reason in E. coli DNA where the replication originates. Such origin are termed as origin of replication. It is, इसे भी आप नोट कर ले, ज़्यादा बेटर होगा. Okay, this is very important for the point of view of examination. Okay, now now because of the requirement of origin of replication that a piece of DNA, if needed to be propagate during the recombination DNA procedure and require a vector, the vector provide the origin of the replication. Further, not every detail of replication understood well. In eukaryotic, the replication of DNA takes place at S phase of the cell cycle, and the replication of DNA and the cell division cycle should be highly coordinated. A failure in a cell division after DNA replication result into a polyploidy and a chromosomal anomaly. You will learn detail nature of origin and the process of occurring at this site and in higher classes. Trans 6.5 transcription. What is transcription? The process कहते हैं ना, the process of copying genetic information from one strand to of the DNA to RNA is termed as the transcription. Okay, DNA से RNA बनना transcription कहलाता है. And here the principle of complementary governs the process of transcription, and except the adenosine complements now forms base pair with the uracil instead of thymine, and however unlike in the process of replication which one set and the total DNA of an organism get replicated. In transcription only a DNA and only one of the strands is copies into RNA. This necessitates defining a boundaries that would be demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during the transcription has the simple answer. First, if both strands act as a template, they would code of RNA molecules with a different sequence. Remember, complementarity does not mean the identical, and it turns if they code for protein, the sequence of amino acid in the protein would be different, and hence one of the segment of DNA would be coding of for two different proteins, and this would complicate the genetic transformation and the transfer machineries. Second, and the two RNA molecules, if produced simultaneously, would be complementary to each other, and hence would form a double standard RNA, and this would prevent RNA from being translated and into protein, and exercise of transcription become a futile one. 6.5.1 Transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA. फर्स्ट प्रोमोटर, स्ट्रक्चरल जीन एंड ट्रिमिटर. इसको काफ़ी पॉइंट ऑफ व्यू से याद कर ले तब मेरे ख्याल से ज़्यादा बेटर होगा. ओके, ये जो है काफ़ी एग्ज़ाम के पॉइंट ऑफ व्यू से ये और ये इसका जो जोड़ा लिखा है इसको पूरा कोई याद कर ले आप तो ज़्यादा बेटर होगा. इसका स्क्रीनशॉट ले ले, आप भी ले लो तो ज़्यादा बेटर होगा. ओके, नाउ वैसे इसका भी हिंदी में भी वर्ड्स में अपलोड कर दूंगा जल्दी. अब बाद में कभी टाइम मिलने पे मैं इसे अच्छे से एक्सप्लेन भी कर दूंगा. अभी फ़िलहाल तो बहुत सारे एग्ज़ाम होने के कारण मैं बिज़ी हूँ, इसलिए मैं इस चीज़ को जल्दी अपलोड नहीं मतलब कि अब सिर्फ अभी के लिए फ़िलहाल रीडिंग ही चलेगा. और किसी को और भी कोई डाउट हो तो यू कैन कमेंट. ओके, आप कमेंट कर सकते हो कमेंट बॉक्स में. ओके, नाउ स्टार्ट. There is a convention in defining of the two strands of the DNA in the structural gene of the transcription unit. Since the two strands have opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 to 3 strands, and that has polarity 3 to 5. Okay, and act as a template and already referred to as template strand. The other strand which has polarity 5 to 3 and the sequence same as RNA and except thymine at the place of uracil is the displaced during the transcription. Strangely, this strand which does not code for anything and is referred to as a coding strand. All the reference point while defining a transcription unit is made with a coding strand and explain the point. A hypothetical sequence from the transcription unit is representing below like this. Okay, template strand and this coding strand. Okay, yes, आगे से पीछे पलट दिया गया और कुछ नहीं है भाई. डरने की बात नहीं खे, समझा. नहीं नहीं मतलब नहीं होता है. इसका भी आप लोग फ़ोटो खींच ले और इस ग्राफ का तो 100% ही ले ले. मैं पूरा ही इसको सजेस्ट कर दूंगा POV ऑफ एग्जामिनेशन. ओके, बहुत काम आएगा. Can you now write the sequence of RNA transcript and from the above DNA? तो इसको आप लोग सॉल्व कर लीजिएगा। नाउ देख लेते हैं। हाँ, the promoter and the terminator flank and the structural gene in a transcription unit. The promoter is said to be located towards 5 to end and an upstream of the structural gene, and the reference is made with the respect to polarity of coding strand. It is DNA sequence that provide the binding site for RNA polymerase and is the presence of promoters in a transcription that unit that also define the template and the coding strand. By the switching its position and the terminator, the definition of the coding template strands could be reserved. The terminator is located towards three end downstream of coding strand, and it is usually define the end of the process of transcription. Figure 6.9. There are the additional regulatory sequence that may be present further upstream or downstream to the promoter. Some of the properties of this sequence shall be discussed and while dealing with regulation of gene expression. 6.5.2 Transcription unit and the gene. A gene is defined as the functional unit of inheritance, and thought there is no ambiguity that the genes are located on a DNA. It is difficulty to literally, okay. Hmm. Define a gene in the term of DNA sequence. The DNA sequence coding for the tRNA or rRNA molecules also define a gene. Okay. However, by the defining cistron as a segment of DNA, एक बात याद रखना, इसका जो सेगमेंट होता है डीएनए का सेगमेंट, उसे हम लोग कहते हैं सिस्ट्रोन। ओके, इसको भी याद कर लेना। ओके, द स्ट्रक्चरल जीन इन अ ट्रांसक्रिप्शन यूनिट could be said to as a मोनोसिस्ट्रोनिक। ओके, ये भी काफ़ी एग्जाम के पॉइंट ऑफ व्यू से काम आ सकता है, इसका भी स्क्रीनशॉट ले लेते हैं। मोस्टली इन यूकैरियोटिक और प्रोसिस्ट्रोनिक एंड मोस्टली इन bacteria and prokaryotic. In eukaryotic, monocystronic structural gene have encrypted coding sequence. The genes in eukaryotic are split. The coding sequence or expressed sequence are defined as exon. Exon are said to be those sequences that appear in mature or processed RNA. Exons are interrupted by the introns. Introns are interview, interviewing sequence do not appear in a mature of processed RNA. The split gene arrangement further complicate the definition of a gene in the term of a DNA segment. Inheritance of a character is also affected by a promoters and regulatory sequence of a structural gene. Hence, the sometimes regulatory sequence and are loosely defined are regulatory genes. Even though these sequence not code for any RNA or protein. 6.5.3 Types of RNA and the process of transcription. In bacteria, there are three major types of RNA and mRNA: messenger RNA and tRNA transfer RNA and RNA ribosomal RNA. All three RNAs are the needed to synthesize protein in a cell. mRNA provides the template. tRNA brings accumulated acid, and RNA read the RNA genetic code, and rRNAs play structural and a catalytic role in the translation. Okay, and RNA is a single dependent RNA polymerase that can be catalyzed transcription of all types of RNA bacteria, and the RNA polymerase bind to the promoter and initiate transcription of initiation. It uses nucleoside and a triphosphate as substrate. इसका भी फोटो आप ले ले। ओके, वेरी इंपॉर्टेंट, सो इम्पॉर्टेंट, सो elegant, just important for exam view. and polymerize in a template dependent on a fashion following the rule of complementarity, if somehow also facilitate opening of the helix and the continuous elongation, and only a short stretch of RNA remains bounded to the enzymes. Once the polymerase reaches to the terminated region and the nascent RNA falls off. Also RNA polymerase the result in the termination of our transcription. And intriguing question is that how is the RNA polymerase able to the catalyze all the three steps, which are initiation, elongation, and termination. The RNA polymerase is only capable to catalyze only the process of elongation. It associates the and transiently with the initiation factor and ठीक है ना, जिसको कहते हैं हम लोग हाँ factor, that is sigma. Okay, and the terminator is small p, यानी कि phi कह सकते हो उसको, and to initiate the terminating the transcription, and respectively association with these factors alter and the specificity of the RNA polymerase to either initiate or terminate figure 6.10 in bacteria, since the mRNA does not require for any process to become active, and also since transcription and the translation takes place in the same compartment, there is no separation of crystol and the nucleus in bacteria. How many the translation can be begin much before the RNA is fully transcribed, and so consequently the transcription and the translation could be coupled in bacteria. In eukaryotic there are two types of additional complexity. There are at least three RNA polymerase in the nucleus in addition to RNA polymerase found in the organelles, and there is a clear cut division of labor and the RNA polymerase first transcribed RNAs. इसका भी फोटो ले लो भाई, बहुत ही एग्जाम के पॉइंट ऑफ व्यू से काम आने वाला है आपके, इसका भी फोटो ले लो। थोड़ा सा दिक्कत होगा बट सहना तो पड़ेगा ना भाई, पढ़ो पूरा बुक पढ़ो, कुछ नहीं छोड़ो बुक का। 20 A's, 18's and 5.8's, where here's ribosome polymerase 3 responsible for the transcription tRNA, 5s RNA, snRNA, small nuclear and RNAs, and RNAs polymerase second transcribed and the precursor of mRNA and the heterogeneous nuclear RNA या HNRNA भी से कहते हैं। ठीक है, भी हट भी जाए भाई। हाँ, नाउ इट्स इनफ। ये सारे चीज़ साइलेंट में है, इसे सही कर दो। नाउ आगे देखते हैं सेकंड। द सेकंड कॉम्प्लेक्सिटी इज दैट द प्राइमरी ट्रांसक्रिप्ट contain both exon and the introns are non-functional, hence it is subjected to a process called splicing. Okay, splicing process भी कहते हैं। splicing क्या होता है जान, स्क्रीनशॉट लेना ये अलग ही चैलेंज है आप लोग के लिए। स्प्लिसिंग क्या होता है, ये पूरा ही जो पूरा दे रखा है ये पूरा ही इम्पोर्टेंट है, इस पूरे को ही समझे। हाँ, splicing where introns are removed and exons are joined in a defined together. In HNRNA undergoes additional process is called capping and tailing. In capping, an unusual nucleotide methyl guanine triphosphate is added to 5 end of HNRNA, and in tailing the adenylate adenylate residue 200 to 300 are the added to 3 end in a template independent manner, and it is fully process of HNRNA and now called mRNA that is transported out of the nucleus for the translation. Figure 6.1. The significance of such complexity is now the beginning to be understood, and split gene arrangement of present and the probably an accident feature of the genomes, and the presence of introns is the reminiscent of antiquity, and the process of simplicity present and dominance of RNA world, and in recent time understanding of RNA and the RNA dependent process in the living system have assumed more importance. 6 point genetic code. During replication transcription, a nucleic acid was copied to form another nucleic acid. Hence, these processes are easy to conceptualize and on the basis of complementary, and the process of translation requires transfer of genetic information from a polymer of nucleotide to synthesize a polymer of amino acid. Neither does not neither does any complementary exist between the nucleotide and amino acid, or nor could be any drawn theoretically. Okay, there existed ample evidence the through to support and the notion that change in nucleic acid genetic material were responsible for the change of amino acid in proteins. This led to preposition of a genetic code that could direct sequence of amino acid during the synthesis of protein, and determining and by the biochemical nature of genetic material and the structure of DNA was very exciting. The proposition of differing of genetic code were most challenging. In a very true sense, it requires involves the of scientists for several disciplines: physicist, organic chemist, and the biochemist, genetics जेनेसिस्ट एंड इन अ जॉर्ज जीनो फिजिसिस्ट। इस ये क्या है, फिजिसिस्ट है। ओके, हाँ, ऑर्गेनिक केमिस्ट एंड द जेनेटिक्स एंड सोन एंड इन इट वाज यहाँ पे, ठीक है ना। It was George Gamow, and a physicist, who argued that since there are only 4 bases, and if they have code for 20 amino acid, the code should be constitute a combination of bases. They suggested that in order to code for all 20 amino acid, the code should be made of 3 nucleotide. This was very bold proposition because permutation combination 4 into 10 into 4 into 4 into 4 यानी कि would generate 64 codon, generate many 4 codon than the require. Providing proof the codon was a triplet and was more daunting task, and the chemical method developed by the हर गोविंद खुराना, ये पूरा किसका दिया गया था, हर गोविंद खुराना द्वारा। Instrumental in synthesize RNA molecules with defined combination of bases, homopolymerase and the copopolymerase polymers, मतलब होता है नहीं, co-polymers, Marshall and the Nirenberg cells free system for protein synthesis finally helped to code to the disferred and the severo Okoa enzymes polynucleotide and the phosphorylase was helpful in polymerizing and RNA with the defined sequence in a template independent manner. Enzymatic synthesis of RNA, finally a checkerboard for genetic code was prepared, which is in a table 6.1. काफ़ी so elegant, so interesting, just important for exam view, नोट कर ले से। ना कुछ इसके साइलेंट फीचर है जेनेटिक कोड के जो कि यहाँ दे रखे। पहले है द कोडोन इज ट्रिपलेट, ओके, and 64 codon of 4 amino acid, and the 3 codons do not for any amino acid, hence they function as stop. Okay, codon. Some amino acid are coded by more than one codon, hence the code is degenerate. The codon is read in mRNA in a centrifugal fashion. There are no punctuation. The code is nearly universal. For example, from bacteria to humans, UUU would code for phenylhine PHE. Some exceptions to this rule and have been found in mitochondrial codons and in some protozoans. 5 number AUG has dual function. It codes for the methionine and mate, and it also acts as initiator codons. 6 UAA are stop terminator codons. If the following sequence of nucleotide is mRNA, predict the sequence of amino acid coded by the take help with the checkboard. टू 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 आगे जान लेते हैं. Now try to the opposite. The following is the sequence of amino acid coded by an mRNA. Okay. Predict the nucleotide sequence in the RNA and the mate phi. Do you know any difficulty in the predicting the opposite? Can you now correlate which two properties are code you have learned? 6.6.1 Mutation and genetic code. The relation between the genes and DNA are the best understood by the mutation study. You have studied about the mutation and its effect in chapter 5, effects of large deletion and the rearrangement in a segment of DNA are easy to comprehend. It may result in loss or gain of gene and so of function, and if and the effect of point mutation will be explained here. A classical example of point mutation is a change of single base pair in the gene for beta globin and chain that result in the change of amino acid and residue glutamate and to valine. It result into a disease condition called as sickle cell amine anemia. And effect of point mutation that insert or deletes a basal in structural genes can be better understood by the following simple example. Consider a statement that is made up of the following, each having three letters like genetic code. Ram has red cup. If we inserted a B in has red and red and rearrange it statement, it would be read as Ram has B, D, K and P. Similarly, if we now insert two letters are in the sample, say BI, now read Ram has beer इडिक ऐप. नाउ वी इंसर्टेड थ्री टुगेदर. देखो इसको इस पूरे को याद ही कर लो भाई साहब, क्या ही मैं बोलूँ इतना, समझ लो इसको, ज़्यादा दिमाग नहीं लगाने का, समझा क्या. Ram has big red cap. The same exercise can be repeated by the deletion of letter R, E and D on one by one and rearranging the statement to make a triplet word. Ram has EDCAP and Ram has DCAP and Ram has cap. The conclusion from the above exercise is a very obvious, and the insertion of deletion of one or two bases changes the reading frame and the point of insertion and deletions. However, such mutations are referred to as framesheet insertion and the deletion mutation. Insertion and deletion of three its multiple base insert or delete in one multiple codon, hence one or multiple amino acid, and the reading frame remain unaltered from that onward point onwards. 6.6.2 6.6.2 and tRNA, the adapter molecule. From the very beginning of the proposition of code, it was clear to Francis Crick. वो भी तो कहीं ना कहीं इंसान ही रहे होंगे. इसका ही फोटो ले लेते हैं. And has to be mechanism to read the code and also link to the amino acid, because the amino have no structural specialist and the read code uniquely. He postulated the presence of adapter molecules and that would one hand and on one hand and read the code on other hand hand would bind to specific amino acid. The tRNA then called sRNA, tRNA को ही बाद में sRNA कहा गया था, इस बात को भी आप ध्यान से समझ ले. Was known before the genetic code was postulated, however its role was as an adapted molecules was assigned much later. tRNA has anticodon loop and that has bases complementary to the code, and it is also called and also has an amino acid and acceptor end to which it binds. Amino acid tRNA are specific for each amino acid and figure 6.12, and for initiation there is another specific tRNA that is referred to as initiated RNA, and there are no tRNA for stop codon. And in figure 6.2, secondary structure of tRNA has depicted that look like a clover leaf, and in actual structure the tRNA is a impact molecules look like a inverted L. Okay. Now translation. Translation refers to the process of polymerization of amino acid to the polypeptides. Figure 6.1.3, figure 6.13, and the order and the sequence of amino acid and are defined by the sequence of base in the mRNA. Amino acid are joined by the bond which is known as a polypeptide bond. The formation of peptide bond which requires energy. Therefore, in the first phase, it himself amino acid are activated in the presence of ATP are linked to their cognate, and tRNA process commonly called is the charging of tRNA and amino cyclation of tRNA to be a more specific if to such in if to such charge tRNAs are about to close, and the formation of peptide bond between would be favorable, and energetically the presence of catalytic would be enhanced, and the rate of peptide bond formation. Then the cellular factory responsible for the synthesizing protein, and it is for the ribosome, and the ribosome consists structural and RNAs have about 80 different proteins, and in its inactive इसमें देखो RNA में होता है 80 अलग अलग प्रोटीन पाए जाते हैं. ओके. And it exists two subunit, a large subunit, a small unit, and the small subunit encounter are mRNA, and the process of translation mRNA protein to begins, and there are two sites there are two sites in the large subunit, and the subsequent amino acid to the bind, and thus it be close enough to each for the formation of a peptide bond. And the ribosome also act as a catalytic, and the 23 sRNA in a bacteria in the enzymes ribozyme, and for the formation of polypeptide bond. And a translational unit in the mRNA is the sequence of RNA that is flanked and by the start codon, and AUG and stop codon, and the code for a polypeptide. And mRNA also has some additional sequence that are not translated and are referred as untranslated region UTR, and UTRs are present at both 5 end and before start codon, at 3 end after stop codon, and they are required for efficient translation process. And for initiation, the ribosome bind to the mRNA at the start codon AUG, and that is recognized only by the initiator tRNA, and the ribosome processed to the elongation phase of protein and the synthesis. And during the stages complexes and the composed of amino acid linked to tRNA sequentially, and thus bind to the appropriate codon is mRNA by the forming complementary bases with the tRNA anticodon. And the ribosome moves from the codon to codon, and along the tRNA amino acid are added one by one, translated into a polypeptide and sequence dedicated by the RNA and the represented mRNA, at the end a release factor bind to the stop codon and terminating translation and releasing complete polypeptide from the ribosome. Okay. Now, regulation of gene and expression. Regulation of gene expression refers to a very broad term and that may occur at a various level, considering that the gene expression result in the formation of polypeptide. It can be regulated at a several level. In eukaryotic, regulation could be exerted and first transitional level and the formation of primary transcript and the processing level, that is regulating and splitting and the translation uh, transport of mRNA from the capsule to the cytoplasm and transitional level. ये सब को भी याद कर ले, थोड़ा ज़्यादा बेटर होगा. इसके टाइप्स को याद कर ले. Now the gene in a cell are expressed to have performed a particular function or a set of function. For example, if an enzyme called beta galactosidase is synthesized for E. coli and used to the catalyzed and the hydrolysis of a disaccharide and lactose into a galactose and glucose. The bacteria use them as a source of energy. Hence, bacteria do not have loose lactose around them to be utilized and for energy, so they would no longer require synthesis of the enzyme beta galactoside, and therefore, in simple terms, it is metabolic, physiological, and the environment or environmental condition that regulate and the expression of gene. The development and the differentiation of embryo into adult organism are also result of the coordinated regulation of expression of several set of genes. In prokaryotic, control of rate of transitional initiation is the predominant site for the gene expression in a transcription unit. In the activity of RNA polymerase at a given promoter is interterm regulated by the interaction with accessory protein which affected to ability to recognize site starts, and regulatory proteins can act both positively and activators and negatively receptors. Okay, मतलब repressors. And the accessibility of promoters region of the prokaryotic DNA is in many cases regulated by the interaction of the protein and with the sequence term operator. The operator region is in adjacent to the promoter element is most operons, and in most cases the sequence of operators to bind repressor and proteins. Each operon has two specific promoters has a specific repressor. For example, lac operators is present only in the lac operon and has interact especially with lac repressor only. 6.8.1 the lac operon क्या होता है? The inclusion of lac operation was also a result of a close association between the genetics and the Francois Jacob and a biochemist Jacob Moe, and they were first elucidate and the transcriptionally regulate them. And the lac operon and had lac refers to lactose, and the polycistronic structural gene is regulated by the common promoters and regulatory genes. Such arrangement is common and in bacteria is referred to as operon. To name few such example, lac operon and trioperon and ada operon and his operon and val operons. ये सब क्या है? operons के example है. Lac operon consists one of the regulatory genes, and that is I gene. Here the I does not mean to inducer, and rather it derives from the word inhibitors. And three structural gene X, Y, Z, sorry, Z, Y, and A. Okay, and I gene code for the repression of lac operons. The Z genes for the beta galactoside and the B gel, and which is primarily responsible for the hydrolysis of the disaccharide lactose into its monomeric units galactose, glucose, and the Y genes code for the premise and which increase the permeability of cell to be galactoside. The alpha gene encode ट्रांस कहते नहीं है transitase, and hence all three genes product in lac operon are required for the metabolism of lactate and lactose. In most other operons as well. The genes present in the operons are need to they together to function in the same or related metabolic pathways. फिगर 6.4 ओके। इस फिगर को भी आप लोग ध्यान से समझ लें, एग्जाम में आने के चांसेस तो नहीं है, बट ये भी आ सकते हैं, कोई कंफर्म तो नहीं है, बट फिर भी अगर रिस्क लेने से कुछ फ़ायदा तो है नहीं, तो इसे आप नोट ही कर लें। ओके एंड नाउ ये तो बहुत है भाई साहब। अभी तक का लॉन्गेस्ट चैप्टर है मेरे ख्याल से, है ना। नो, अब आगे देख लेते हैं। लैक ऑफ लैक्टोज इज द सबस्ट्रेस फॉर द एंजाइम्स गैलेक्ट beta galactosidase, and it regulates the switching and on and off of the operons. Hence that it is termed as inducer. In the absence of a preferred carbon source of such as glucose, if lactose is provided in the growth medium of bacteria, the lactose is transported into a cell through the action of permease. Remember, a very low level of expression of lac operand has to be represented in the cell all time. Otherwise, lactose cannot be intercell. The lactose then induce the operons in the following manner. The repressure of the operons synthesized and all the time of constitutively from the I gene. The repressor protein bind to the operators and the region of the operons and the prevent RNA polymerase and from the transcriptionally the operons. In the presence of inducer and such as lactose and the lactose repressor and inactivate by the interaction with inducer, and this allow the RNA polymerase and access to the promoter and the transcription proceed, and the figure 6.6 and its essential regulation and the lac operation can also be visualized as regulation of enzyme synthesis and its substrate. Remember, glucose and the galactose cannot be as a inducer and lac operans. Can you think how long lac operans can be expressed in the presence of lactose? Regulation of lac operon by the repressor and to as negative regulation. Lac operance is under control of positive regulation as well, but it is beyond scope of discussion of this level. तो आगे आते हैं मैंने आगे ह्यूमन जीनोम प्रोजेक्ट। In the preceding section, you have learned that it is the sequence of the base in DNA that determine the genetic information of a given organism. In other words, the genetic makeup of an organism or an individual lies in the DNA and sequence. If two individuals differ and their DNA sequence should be also different and at least some places, the assumption to lead quest of finding out the complete DNA and the sequence of GNA humans genomes, and with the establishment of genetic engineering technique where it was possible to isolate and the clone any piece of DNA, and availability of simple fast and technique for determining DNA sequence, a very antibiotic project of subsequencing human genome was launched in the year 1990. Human Genome Project HGP was called मेगा प्रोजेक्ट। HGP को हम लोग मेगा प्रोजेक्ट भी कहते हैं। And you can imagine the magnitude, the requirement for the project, and if we simplify define the aim of project as follow. Human genome is said to be have approximately 3 into 10 power 9 BP. Okay, if the cost of consecuting required is $3 per BP, estimate in the beginning. The total estimate cost of project would be approximately 9 billion US dollar. Further, if the obtained sequence were to be stored in the type of form in the book, and if page of book contained 1,000 letters and in each book contained 1,000 page, and then 3,300 such a book would be required to store the information of DNA sequence and from human cell. Enormous amount of data expected to be generated also necessitate the use of high speed and the computational and devices for data storage and retrieval analysis. HDPR was closely associated with the rapid development of new area of biology is called bioinformatics. Okay. ह्यूमन ग्रोथ जीनोम जो होता है उसका भी goals of HGP. Some of the important goals of HGP are as for what was follow. Identify first identify all the approximately 20,000 to 25,000 genome in human DNA. Second, determine the sequence of the 3 billion chemical base pairs that make up human DNA. Third, store this formation of database. Fourth, improve tools for data analysis. Fifth, transfer related technology to other sectors such as industries. Sixth, address ethical, legal and social issue ELSSI and that may arise from project. The Human Genome Project was a 13 years project coordinated by the USDR Department for Energy and the National Institute of Health during the year of the HGP, and the Wellcome Trust UK and the became the major partner. Additional contribution came from the Japan, France, Germany and the others. The project was completed in 2003. The knowledge about the effects of DNA variation and among individual can lead to evolutionary and new ways to diagnose, treat, and someday present the thousands of uh, disorder that affect the human beings. And beside the providing clauses to understanding human biology and learning about non-human organism and DNA sequence can lead to be understanding and their capabilities that can be applied towards solving the challenge in healthcare and agriculture, energy production, environmental remediation. Many non-humans model organisms such as bacteria, yeast, and the and कहते नहीं है cyanide habitats and illegence of free living and non-pathogenic nematodes, Drosophila, a fruit fly, and the plants, rice and Arabidopsis and etc. have a sequence. Myth, कहते हैं methodologies. The method evolves to measure a process and the approach focus identifying all the genes that are expressed as RNA and refers to an express sequence tag etc. And the book and the took behind approach and for the simply sequencing and the whole of set of gene and that combine that contained all the coding and non-coding sequence, the later assigning different region in the sequence with the falser and a term to as sequence annotation. Okay, and for sequence the total DNA and from a cell is isolated converted to a random fragment of relatively smaller sizes. And recall the DNA is a long polymer and there are technical limitations in sequencing very long piece of DNA and clone in suitable host using specialized vector. The cloning result into an amplification of each piece of DNA fragment, and so that it subsequently could be subsequent and with ease, and commonly used to host and were bacteria and yeast. The vector were called BAC, bacterial artificial chromosome, and YAC, yeast artificial chromosome. This is very important. The fragment were sequenced using automated DNA, DNA sequence, and that work on the principle of method developing by the Frederick Stranger. And remembered Stranger is also con- contracted for the developing method of for determination of amino acid sequence of protein. And these sequences were arranged based on the overlapping region present in them, and this required generation of overlapping fragments of for sequence. Alignment of these sequences are humanly not possible. Therefore, specialized computer based on programs were developed. Figure 6.5, and these sequences were subsequently annotated and were assigned to each chromosome, and the sequence of chromosome I was completed in May 2006, and this was the last of the 24 chromosome and 22 autosome and X and Y. यस ये इसको देख लीजिए ओके इस ये भी काफ़ी इम्पोर्टेंट हुआ बट ये उतना इम्पोर्टेंट लग नहीं रहा है फिर भी आप इसको देख लीजिए देखने में क्या ही जाता है आई थिंक ये सबसे लंबा ही चैप्टर था अभी तक का बहुत लंबा चैप्टर है ये भाई साहब the sequence. Another challenging task was assigning and the genetic and the physical map of genomes, and this was generated using formation of polymorphism or restrictive endonuclease and recognition sites, and some of repetitive DNA and sequence known as the microsatellite. And one of the application polymorphism, and it repeat, repeat repetitive and DNA sequence shall be explained in the next section of DNA fingerprinting. 6.9.1 Salient feature of human genome. Okay, some of the salient feature observation drawn from the Human Genome Project are as follow. First, human genome contain 3164.7 million pp, and second, average gene consists 3000 bases, but sizes vary greatly, with largest known human gene being di- dystrophin at 2.4 million bases. Third number, the total number of genes is estimated at 30,000, much lower than previous estimated of 80,000 to 1,40,000 genes. Almost all 99% nucleotide bases are exactly the same in all people. Fourth, the functional are known of over 50% of the discovered gene. The less than 2% of the genomes for proteins. Repeat sequence make every large portion of the human genome. Repetitive sequence are stretches of DNA sequence that are the repeated many times, sometimes hundred of thousand times, and they are taught to have no direction coding function, but they shed light and or chromosome on chromosome structure dynamic evolution. Chromosome I has most genes 2968, and Y has the fewest 231. इसको अपने फोटो खींचा नहीं होगा इसलिए scientists have identified about 1.4 million location were single base DNA differences. SNP single nucleotide polymorphism, pronounced as a SNP, and occurs in human, and the information promises and revolutionize the process of finding chromosomal location for the disease associated subsequent and the tracing human history. Now, application and the fun's future challenge. Deriving meaningful knowledge from the DNA sequence will define research through the coming decades and leading to our understanding biological system. The enormous task will require the enterprise creativity of 10,000 of scientists from varieties and discipline in both public and private sectors worldwide. One of the greatest impact having HG sequence and may well be enabling a radically and new approach to biologically research. In the past, research studied one or a few genes at a time. With whole gene sequence and new high throughput technology, we can approach question to systematically and on a much broader scale. They can study all the gene in a genome and for example the transcription in a particular tissue or organ or a tumor, or how tens of thousands of genes and protect proteins work together in interconnection and network orchestrate the chemistry of life. DNA fingerprint 6.10. As stated in the preceding section, 1990% of bases and among humans in the same. Okay, assuming the human genome as 3 into 10 power 9 bp in the many base sequence would be there be differences, and it is these differences in sequence of DNA which make easy individual unique in the phenotypic appearance. And if one aim to be find out genetic differences and between the individual or among individual of a population, and sequencing the DNA every time would be daunting and expensive and the task. Imagine try to compare two set of 3 into 10 about 6 base pair. DNA fingerprinting is a quick way of compare of DNA sequence of any individual. DNA fingerprinting individual identify the difference in some specific region in DNA sequence is called repetitive DNA. Okay, and because in this sequence a small stretch of DNA repeated of many times, the repetitive DNA separated from a bulk genomic and DNA as the different peak during the density of gradient centrifugation. The bulk DNA forms a peak, and other small peak are referred to as satellite DNA. And depending on the base composition and the length of segment and the a number of repetitive units, and satellite DNA is the classified into many categories such as a microsatellite, mini satellite, and this sequence normally do not any protein. Okay, code for protein, but they forms a large portion of human genomes, and this sequence so high degree of polymorphism and form the basic basis of DNA fingerprinting. And since the DNA are from every tissue such as blood, okay, such as blood, hair follicle, and skin, bone, and saliva, sperm from an individual show the same degree of polymorphism, and They become a useful identical tool in the forensic application. Further, as the polymorphism inheritable from parents, DNA fingerprinting is the basic paternity testing in case of dispute. As polymorphism in DNA sequence is the basis of genetics mapping and of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism mean. In simple term, polymorphism variation at genetic level arises due to the mutation and recall different kind of mutation and their effect that you have already studied in chapter 5. Okay, and the preceding section in the chapter. New mutation may arise in an individual either in somatic cell or germ cell, cells generated gamete and in sexually reproduction organism. In a germ cell, mutation does not seriously impair individual's ability to have offspring and who can transmit the mutation, and it can spread to the member of population and through the sexual reproduction. Okay, उसके बाद क्या है? Allelic. Allelic, again recall the definition of alleles from chapter 5. The sequence variation and has traditional has been described as a DNA polymorphism, and if more than one variant allele at a focus locus occur in human population with frequency greater than 0.01. Okay, and in simple terms, if an inheritable mutation is observed in a population at high frequency is referred to as NDA polymorphism. The probability of such variation to be observed is an non-coding DNA sequence would be higher. Mutation in this sequence may not have immediate effect and impact in individuals and reproductive health ability. Mutation keeps on accumulating a generation after generation from one of the basis of variability and the polymorphism. There is a variety of the different type of polymorphism ranging from the single nucleotide change to life cycle, large cycle, largest change, large scale changes, and for human for the evolution and the speciation and the polymorphism play a role, very important role, and you will study this detail in higher classes. And the technique of DNA fingerprinting was initially developed by Alec and Jeffrey, and he used a satellite DNA as a probe that shows a very high degree and polymorphism. It was called as variation number of tandem repeats and wear and tear. At the technique as used earlier involved south blots and the hybridization and using radio labeled VNTR as a probe. First, isolation of DNA, digestion of DNA by the resistance of Indian nucleus, separation of DNA fragment by electrophoresis, and transferring blotting of separate DNA and the fragment to synthetic member and such as nitrocellulose or nylon. Hybridization labeled using labeled VNTR probe and detection of hybridized DNA fragment by the auto radiography and a schematic and representation of DNA fingerprinting is shown figure 6.1.6. VNTR belongs to a class of satellite DNA referred to as a mini satellite, and a small DNA sequence is arranged of randomly in and many copy number, and the copy number varies from the chromosome to the chromosome in an individual. The number of ripples show a very high degree of polymorphism as a result of the size of VNTR varies in the size of from 0 to 1 to kb 0.1 to 20 kb. Sequentially, after hybridization, VNTR probe the auto radiogram and gives the my brand of differing size. This brand give us characterizing pattern of for an individual DNA figure 6.6, and it differs individual to in a population, except in the case of monozygotic and identical twins, and the sensitivity of technique has been increased by the use of polymerase chain and the PCR. You will study it about in the chapter 11th. And consequently, DNA from single cell is enough to perform DNA fingerprinting analysis, in addition to application of forensic। इस बॉडी चीज को ध्यान समझ ले तो ज़्यादा बेटर होगा। Forensic science, and it has much wider applications such as determining population and genetic diversity, and currently many different probes are used as to generate fingerprint। Summary: nucleic acid are long polymer of nucleotide, while DNA stores genetic information and the RNA mostly helps in the transfer and expression of formation through the DNA, and RNA function as genetic material, but DNA being chemically structurally more stable is better genetic material. And however, RNA is the first evolved and DNA was derived from RNA, and the hallmark of the double-stranded DNA helical and structure of DNA is the hydrogen bonding between the bases of from the opposite strand, and the rule is that the adenine pair with the thymine, and 2H bond with and guanine cytosine with 3H bond, and this make one strand complementary to the other, and DNA replicates semi-conservatively, and the process of guided by the complementary H bond, and the segment of DNA that the code for the RNA is in simplistic and term can be referred gene, and during the transcription also one of the strand of DNA act as template direct and the synthesized complementary DNA, and in bacteria transcripted functional, and hence can directly to be translated in eukaryotic gene is split, and the coding sequence exon are interrupted by non-coding sequence introns, and introns are removed, exons are joined to produce functional RNA by the splicing, and the messenger RNA contain base consequential, and that are read in a combination of three and the to make triplet genetic code to the code of amino acid, and the genetic code is read again on the principle of complementarity, and that by the tRNA that act as an adapter molecule, and that there are specific tRNAs of every amino acid. The tRNA bind to a specific amino acid at the end, and the pair through the H bonding with code on mRNA and through its anticodon, and the site of translation and the protein synthesis and is ribosome which bind to RNA and provide to platform and joining of amino acid. One of the tRNAs act as a catalytic for peptide and bond formation, which is an example of RNA enzymes. Ribozymes and translation is a process that evolves the around RNA and indicating the life of RNA, since the translation are genetically expensive process, and these have to be tightly regulated, and regulation and transcription is the primary step for regulation of gene expression. In bacteria, more than one is arranged regulation and Hmm, regulation and is primary step for the regulation gene. In bacteria, one gene is arranged together and regulate unit called operons, and lac operons is the pro- prototype operon and in bacteria, which code for gene responsible and metabolism of lactose. Operons regulate the amount of lactose and medium and where the bacterium are grown. Therefore, regulation can also be viewed as regulation of enzyme synthesized by substrate. Human Genome Project was a mega project that aimed to sequence of sequence every base in human genome. The project was yield much new information. Many new areas and avenues have opened and us up as a consequence of project. DNA fingerprinting is a technique to out variation in individual of a population at DNA level. It works on principle of polymorphism is a DNA sequence. It has immense application in the field of forensic science and genetically biodiversity and evolutionary biology. So guys, thanks for वॉचिंग द वीडियो टिल द एंड। बट आपका थोड़ा सा फर्ज बनता है इस वीडियो को लाइक कर दे, क्योंकि वीडियो बहुत लंबा है और ये चैप्टर इतना ज़्यादा तक कंटिन्यू रीडिंग करने में बहुत टाइम लगता है। ओके, सो आपको ज़्यादा कुछ नहीं, बस लाइक एंड सब्सक्राइब करना है और यू आर अ पार्ट ऑफ जर्नी ऑफ नीट, यू नो एज वेल एज। सो बाय एंड सी यू इन नेक्स्ट वीडियो, एंड इसको जितना हो सके उतना शेयर भी कर देना। बाय।